Hello, good evening, everyone. First of all, thank you so much for coming to this Pint of Knowledge. As many of you would know, we have here, this is the fourth Pint of Knowledge. We have had people from different backgrounds presenting their research, such as astronomy or biology. So if you are interested in participating, volunteering, just please, contact us either in our social network, sending us an email, or just talking to us directly. And today it is the turn for medicinal chemistry. Albert has been working there in the Department of Chemistry at the Spring Group of the University of Cambridge. We'll be talking, and now he has moved to the Pharmaceutical Industry. We'll be talking about the exciting world of the chemistry of peptides. So I'll leave you with him.
Laura, thanks for the introduction. Thanks. Okay, so before diving straight into the world of peptides, let's talk a little bit about proteins. So what is a protein? Proteins are large biological molecules, and we have them in our body, and they play very important roles. And their constituents are one or more chains of amino acids. And there are about 20 main amino acids for proteins. And playing so many important roles in our body, they are common targets for medicines, right? So what is a peptide then? A peptide is just a shorter chain of amino acids, up to 60. You can see one of them here. And sometimes are represented with these little helix, right? Okay, so just as a reminder, an amino acid is this here. I don't know if you can see it properly. The main thing is that it has a naming group, it has a carboxylic acid group, and also it has a side chain which is different for each of the 20 amino acids. When this amino acid is part of the protein, you can see it here, we call it amino acid residue. It's just the same amino acid minus water, and you can see it in here. It's part of the protein. Right. Okay. Now we know what peptides are, but these are some of the other questions I'll try to answer during this talk. So first thing is, what makes peptides useful as medicines? Then what's medicinal chemistry? Then where can we find these peptides? We'll talk also about the ideal drug, and by drug I mean medicine in this case. Then we'll see if we can get peptides into human cells. And I'll talk a little bit about the future of peptide medicines. Right, so peptides are useful as medicines. There are about 100 peptides that are in the peptide-based drugs or medicines that are currently on the market. And that's about 25 billion per year annual sales. Right? And they can treat the so-called big diseases like metabolic diseases such as diabetes. They can also treat cancer, HIV, bacterial infections, and chronic pain, among others. A typical example is the peptide T20 that you can see here. It's quite a big thing. So each letter represents an amino acid. So it has 36 amino acids, and the molecular weight is more than 4,000. So if you compare it with the typical medicine that we're taking, like paracetamol has a molecular weight of just 151. So peptides are generally bigger than the most common medicines. And the most important thing of this slide is that recently there's been an increasing number of peptidic medicines in the market. Okay, so if you want to know a little bit more about this, you have to wait a little bit, because in the next slide I'm going to tell you where we can find peptides. Right, so where can we find them? A lot of them are natural products. So we can find them in the sea, we can find them in the deserts, we can find them in the forest, in many places. So these are two examples I've chosen. This one is aplidine, it's a cyclic peptide, so the amino acid chain but just a cycle, and it's in this sea organism, and is currently in trials as an anti-cancer drug. Right, so these other peptides I've chosen, sorry, you can see very well, it's just another beautiful marine organism. the corn snail, and scientists have discovered recently in March 2014, so a few weeks ago, that they can use these peptides to. They are a possible treatment for the chronic nerve pain. Right? But if you don't like scuba diving, don't worry, you don't have to go that far away to find peptides, because we can find them in our body. So the famous endorphins that our body generates when we do something pleasant, like running, for instance, they are peptides as well. They are linear peptides. You can see one of them here. And the other way of getting peptides is just asking a chemist to make one for you. And sometimes we even get a PhD for making them. Right. But how do we make them? So that's a dipeptide. It's the simplest peptide, just 2 amino acids. And the way to make them would be mixing these 2 amino acids with some other chemicals and then see if we obtain our dipeptide. So what happens is that this acid and this amine here, they react, they form this amide bond here, or peptide bond, we call it as well, and we lose water. The problem is that we have this free amine and we have this free acid that can react as well, so then we end up with messy mixtures of polypeptides. And so what can we do about it? So what we do is we use protecting groups, these species, they are to protect the amines, to protect the acids as well, and to protect the side chains of some amino acids. These protecting groups are just chemical molecules that we chemists design. And if we protect the amine, we protect the carboxylic acid of the other amino acid, only this carboxylic acid and this amine are free to react to form the amide bond, and then we get our protective peptide, and the only thing we have to do then is remove chemically these 2 protecting groups and get our product. So that's how we make peptides. Well, that's how we make a dipeptide. What if we want to make something bigger, like the T20? Well, if we do it by solution chemistry, I mean the typical round bottom flask with mixing some chemicals in a solvent. What happens is that amide bond formation here, growing the chain. The problem of this scheme is that we have to isolate these 2 compounds before.
We have to isolate these before continuing. So we have to do a work to separate it from the solvents and from other impurities. And then we have to do a purification to remove other impurities that we have there. So I don't know if you can see it properly, that's just part of the workup. And that's just the evaporation of the solvents. And that's the purification in which you can see the different chemicals here. So these steps take a long time. So they take hours, each of them. So if we're making a dipeptide, it's a few hours, that's all right. If we're making the T20, so for a dipeptide, it's just 2 workups, 2 purifications. We do it like that, it's fine. For the T20, is 36 amino acids, 70 workups, 70 purifications, a really long time. So we can't really make it this way, right? So then is when solid phase synthesis came. So Bruce Merrifield won the Nobel Prize of Chemistry in 1984. And in the 60s, he decided to do peptide chemistry by attaching the peptide to a polymer, an insoluble polymer or resin. In his case, it was polystyrene that you can see here. That's a microscope photograph. And you can see a representation here. So polystyrene, we have it. We've seen it quite a lot. For instance, the CD cases that are made of polystyrene. You can see that the white beads and the colorless bead are both polystyrene. So depending on what we do to it, we can give it different consistency. And in this case, for the polymers or resins, is very flexible and is spherical, right? So for it works is you have your polystyrene bead, then you take your protected amino acid, attach it to it, make it react with it with some reagents, and then you have a syringe here with a filter. That's what I was doing last week. So you have a syringe here, there's a filter here, and there's a tap here that you keep close while the reaction is going on. So that reacts, the amino acid gets attached to the support, and then to get rid of all the impurities and the solvent, what you do is just a quick filtration and some washings. So what before took a few hours, now it just takes filtering and washing for 5 minutes, and it's on. Then you just have to remove the protective group, which you do, and again you just filter,
the polymer of polystyrene is insoluble, so it stays there with the amino acid, and you get rid of all the impurities. Then if you want to make something longer, you repeat this thing quite a few times, and when you're done, when you have your peptide sequence, you just do a final cleavage from the support. So you just break this bond here, and you got on one hand the polymer here, and on the other hand the peptide, which is usually a white solid. Right? Okay, so more or less at the same time as Merrifield, similar time, there was this guy, Richard Houghton, so he decided to use T-bags instead. So he's not English, he's American, but he decided to use T-bags. What the guy did is instead of putting tea in the tea bag, he just put polystyrene. So it's a different option. So he put polystyrene inside this, and then what he was doing is just soaking this tea bag into an amino acid solution. Then the amino acid got attached to the polystyrene in the bag, but it was still dirty of other reagents and amino acids. And then he just did some washing, just again put the tea bag somewhere, wash it, and get the amino acid, and then if he kept going, he would get a peptide. Okay, we're not using tea bags now, we're using these syringes, but we're doing a similar thing, right? So, but what chemicals are we using for these to do reactions? So, I'm not going to talk about the amide conformation. I'm going to talk more about the removal of this PG or protecting group and the final cleavage from the polymer, right? So what we use here, or what Merrifield used to use, is this Bob benzyl chemistry. Don't worry about the name. The important thing is to know that the PG or protecting group is Bob, and it was removed with an acid, with trifluoroacetic acid. And then to do the final cleavage from the support, they use a more reactive acid, which is hydrofluoric acid. This hydrofluoric acid is a very, very clean agent. It works really well. It's volatile. You can get rid of it. The problem is that this is a little bit dangerous. So I don't know if you've seen Breaking Bad, but in the second chapter, they try to get rid of some corpses just to dissolve them, and what they use is hydrofluoric acid, which I'm surprised it was in a high school, but yeah, they use it. And the guy makes a mistake, and instead of using a plastic recipient, he used a bath tube. So that's what he did to the bath tube, and that would happen in reality. And it will probably be worse. So yeah. So hydrofluoric acid is better if we can avoid it. So what we do now is using this mock-butyl chemistry. Important things here is that we use a base to remove the protecting group, and to do the final cleavage we use a trifluoroacetic acid. So the advantages here is that it's milder, so that's not going to happen to us. That's good. The other advantage is that it is orthogonal. That means that we're using different types of reagents to do the two chemical reactions. We use a base and we use an acid. So then everything is going to be more selective. In this case, it's like if you heat something softer to remove the bulk, and then you heat something really hard to remove to do the cleavage. So when you do it softer, if you do it many times, maybe you can still have some cleavage, so it's not that selective. So what we use now is this second one, that mock-butyl chemistry. So I don't know if you can see it very well. That's a manual system that I'm using now. So in general, you use the manual system for doing complicated reactions on the peptide or for expensive building blocks. So these are these are two peptides. These two work really well. This one didn't work, but yeah, it's fine. Then the important thing here is that we can do parallel synthesis. So we can have these three syringes. This green thing is a stirrer. So you just stay there, stirring, and you can do it with two hands. It's kind of parallel, you can change hands. Okay, so that's a manual way, but now we have more sophisticated things as well. So we can do it automatically. So that's an automated synthesizer at room temperature. So here you can put a few reactions in parallel, about 12 reactions, 12 peptides. And here you put your amino acids, your amino acid solutions. There are more than 20 positions because you can use non-natural amino acids, etc. So yeah, that's pretty good. What is this one? This one is in this one you can do parallel chemistry. It's just one peptide that you can make at the same time, and that's a microwave synthesizer. So the principle is the same as the microwave at home. So, but it's a little bit more sophisticated. You have the microwave here, and you have this syringe or something like that. You have it inside this, and here you have the amino acids. And it's just for complicated reactions that need an extra kick. So with this machine, you really make them to work, right? So, well, that's another challenge we find in peptide chemistry, and it's quite important. So it's chirality. Okay, what is chirality? First, what is not chirality? So achiral means something that is identical to the mirror image. So if you look at this, there is a cube, and that's a mirror, and behind the mirror we have exactly the same thing, right? So that's achiral, right? Okay, chiral things are these things that are different from the original molecule. So you have a molecule that's different from the mirror image is different from the original molecule. So if you put your hand and you have a mirror, behind the mirror, so if you have the left hand here, you're going to have the right hand behind the mirror, right? And they are not the same. So they interact very differently with different things. So if I try to shake the hand to someone, if they don't offer me the same hand, it's not going to work. That's important. So most amino acids are chiral, most of them. So that makes most peptides chiral as well. So we know that a peptide is a chain of amino acids, so a chain of hands in this case. So if you want to get it right, we don't only have to get the right color code. So each color represents an amino acid. We don't only have to get right the color sequence, but for each amino acid, we have to get the correct hand, right? And that's really important because if we get only one of them wrong, our medicine might not work. That's the best case scenario, or it might be toxic. So we have to be really careful. But we have tools, we have chemical tools to do that, and we have analytical tools to analyze our products and see that we got the right thing. Right? Okay. So that's a peptide I made during my PhD. It's an anti-tumoral peptide, and you can find it in the sea as well. So that's just so that you see that sometimes you need 2 protecting groups, sometimes you need 3, sometimes you can need even 4 or even more. In this case, we have a cycle here, and we want to functionalize this bit, and this bit can react as well. So we need to protect the different bits that can react, and these protecting groups need to be orthogonal. So we should be able to remove each of them without affecting the others, right? And these protecting groups is we chemists that we designed them. They are small molecules, and that was part of my PhD as well. Okay, let's go back to peptides and medicines. Why are peptides useful as medicines? Well, let's wait a little bit. Before, what's the ideal medicine or drug? So some of the things it needs to have, it needs to be potent. So we want to have a low dose of it, and we want to have it if possible once a day, not like 3 times a day. The other thing is that it has to be selective. So we don't want side effects. We don't want something for the stomach ache that causes us headache. It has to act when it has to act and nowhere else. Then it needs to have good pharmacokinetics. That means that pharmacokinetics is what the body does to the medicine, right? So the medicine, before reaching the place where it has to act, many things can happen to it. It can get degraded, for instance. It can get excreted before doing anything. So we need good pharmacokinetics. And the other desirable thing is that the medicine is oral. So most of us prefer having a little pill than having an injection. So that's something that we prefer on medicines. Okay, so what do medicinal chemists do to try to obtain medicines? Well, the definition is apply the chemistry to the discovery of new medicines. Okay, but what does this mean? That means that we synthesize some molecules that can be potential medicines. That means that we design these molecules as well, so that they are likely to be good medicines. And yeah, to do that we use computational chemistry and many other tools. And then something really important in drug discovery is that we collaborate with other scientists, mainly biologists, but physicists as well, and many other scientists, and that's really important, right? Okay, so why are peptides can be good and are good as medicines? So most medicine targets are proteins, right? Some peptides are made of the same building blocks as proteins. So they're going to interact really well with them. They're going to have high affinity with them. Then they're going to be selective as well. They're going to be peptides are non-immunogenic. That means that our immune system, when it finds a peptide, is not going to react and really go for it. And they are relatively easy to make, as you've seen. They have also some limitations, but yeah, we'll talk about them later, okay? Okay, so I said that the main targets for medicines are proteins, but we can target one protein, which has been the usual approach until now, but we can also target interactions between proteins. So a lot of biological processes in our body work because two or more proteins are just interacting and they have to be together. And some diseases also happen because two or more proteins interact. So if we target these protein-protein interactions, there are many of them. Some people say it's about 80% of the drug targets. Well, I don't know, it's a lot. Let's stay with them. And most of them are inside the cell. So if we target these protein-protein interactions, we can cure a lot of diseases. The problem is that these protein-protein interactions are poorly tractable with the small molecules, with the typical medicines that we have now. So there is a need for new types of medicines, and this is where peptides come. Okay. So a little bit about protein-protein interactions. We have a protein here. You can see, I don't know if you can see it properly, that's an alpha helix. That's a helix. It's a secondary structure of the protein. And this helix is present in a lot of protein-protein interactions. Right? So here you have an interaction between proteins. And you see that this bit that's interacting is a helix. So can you see this thing here? I don't know if you can see it properly. I can hardly see it from here. So that's what is a typical medicine that we're taking now. So what we've been trying to do is trying to make this tiny little thing to disrupt this massive thing. Okay, you can do that sometimes, but usually that's not working. So you need something larger to disrupt this protein-protein interaction and to cure the disease. So you can try different approaches. One of them is antibodies, and they work really well, but I'm not going to talk about this. The other approach is obviously peptides. So if you mimic this helix with a peptide, so you just synthesize the sequence and do something else that we'll talk about later, and add it to your protein-protein interaction in the body, this peptide can potentially just displace the massive protein and then generate a cure for this disease. Right? That's roughly how it works. But what are the historical limitations of peptides that before I mentioned a little bit whispering? So one of them is that they're not very stable in our body. So we have some enzymes called proteases that the moment they see a peptide in the wrong place, they just eat it, they just degrade it, and you get a few amino acids, and these are not going to be useful as medicines. The other limitation is also peptides have low cell permeability. In the last Pint of knowledge, we learned a little bit about cells. That's a cell. That's just a cartoon of a cell. So you see that you have this membrane here, right? So this membrane is a bilayer of lipids, so fat, and it has also some proteins. So basically, when the peptide reaches this membrane, doesn't know what to do, and it simply doesn't cross it, and it can't interact with the proteins, right? So these are two historical limitations. What are we doing about it? Right. So in terms of the stability, we managed to make stable peptides. For instance, if we make a cyclic peptide, it tends to be much more stable because the protease doesn't know where to start eating it. That's one of the principles. It's more complex, but it's roughly the idea. Then an example of this is cyclosporine, which is an immunosuppressant peptide that we use in organ transplantation. Then another way is staple peptides. Remember this name, we'll talk about them later on. Then you can use non-natural amino acids. So you can put some non-natural amino acids here. This way you will confuse the proteases of your body, and they won't break the peptide. Something else you can do is you can make peptidomimetics, and that's something I did during my postdoc here. You can just put things that are not amino acids inside the peptide. Again, protease will be really confused, and they will be stable. And then you can do many other things. One of the things, for instance, is conjugate it to another molecule, sometimes bigger, that would protect it from the proteases, right? So this stability issue is solved for a lot of peptides. Not for all of them, but for a lot of them. Right, what about cell permeability? Here is not solved yet, it's not completely solved. It's solved for a lot of peptides, but not for all of them. Some strategies that we use, we can mask the polarity, so we can hide the things that are soluble in water and put the fatty things into display, so that these peptides can cross this fatty membrane. Okay, that's a strategy, and that works sometimes. The other strategy is again about staple peptides that I'll talk a little bit more about later. But for now, just to know that a p53 peptide is in the clinic, is in clinical trials for cancer. So these guys are getting there. Then you can use molecular shuttles. This one example of this is the HIV TAD. The HIV TAD is a short peptide that is in a protein of the HIV. And this short peptide, this one is cell permeable, is responsible for the HIV to get into cells. So if you attach this fragment to another peptide that's not cell permeable, sometimes you can make it just cross the membrane as a shuttle. Other things you can use are delivery technologies. There are a lot of them, but this is not my topic again, so just believe that we can use them. And you can do many other things. So we're not there yet, but we're really getting there, right? Okay, so what about staple peptides? They appear twice, I didn't even say what they are. Okay, so first the concept of entropy. So entropy is a tendency to disorder, right? So here you can see, you can kind of see my desk. So yeah, after a little bit of time, what happens is that it becomes like this. So the same can happen to molecules, right? So that's a protein, we've seen it before. So it has here this alpha helix that we've seen before. So if we try to mimic this alpha helix with a peptide, we just synthesize this little sequence here, hoping that it stays alpha helical when it disrupts a protein-protein interaction. We're going to have entropic effects, and that's going to happen is this. And this is not going to interact with our proteins, and it's not going to be useful. So what Verdine and company decided to do is use a chemical staple. So it's just a staple. It's not a metallic staple. It's a different thing, but the idea is the same. It's just stapling these two positions so that there's something that's holding the helix together. In the protein, what's holding the helix together is the whole protein, right? In here, we have this staple that a lot of times it manages to hold the helix and it manages to improve the properties of the peptides. You have to be really careful where you put the staple, but a lot of times it works well. Right? Okay. So, to conclude, we've seen that peptides can be useful as medicines. We've seen I've said that they have high affinity to proteins because they are made of the same building blocks. That they are selective as well. They are non-immunogenic, so they don't generate an immune response of our body. They are easy to make, and most importantly, they can disrupt these protein-protein interactions that are so important to discover in medicines. Then, about the limitations, there are multiple novel chemical strategies to solve the stability and cell permeability issues, mainly the stability, but also the cell permeability. And there are increasing numbers of peptide therapeutics, about 100, but there are about more than 400 that are kind of getting there and about to make it. Right? So the conclusion is that is looking good for peptides, right? Okay, so with that I'm done, so I'll be really happy to answer any questions you have.
I have a question.
Done.
If you can go back to
Yeah.
where the amino acids bind to the resin,
Yeah.
just before the one with the tea bags.
It's the one.
Yeah. So I really don't get how you actually do the whole binding between the
Yeah.
You just add them together to the resin and they just bind together. Like
No, you have to add some reagents here. So what you do is so what you do is see it here. So you add some reagents to activate this guy. So if you mix them together, they're not going to react. So you need to activate this acid, and then this amine can just attack it, and water goes, and you just get your molecule. So you need to add some reactions in there.
And if you're using lots of amino acids to do a big peptide, as you're doing on the next slide,
So
then
yeah,
you just add them all the amino acids together.
yeah, no. So what you do? Okay. So what you do is you do one by one, right? So for the first amino acid you want in your sequence, you add it here, protect it with the reactions that will activate it. Then it reacts with an amine that's on the resin, right? Then it's linked to the resin. Then you get rid of all the solvents and everything. You remove your protecting group, again wash. Then you have one amino acid. So then what you would do is with this, go back to a style. You add here another protected amino acid. You have the amine here, so that's going to react. You do the washings. You remove the protecting group, do the washings again, and then repeat this cycle quite a few times with different amino acids each time, and then you get your paper. It depends. That's a good question. So sometimes it's okay doing it once and doing it for 20 minutes, for instance, a coupling, and the protection can last 10 to 15 minutes. If you do it in the microwave, for instance, you can do it 3 minutes or 5 minutes, this coupling, and then 3 minutes for this protection, more or less. So in the microwave, for instance, in about 2 hours, you can make maybe a 10 amino acid peptide or 20, depends. With a manual, with a room temperature, I don't know, 24 hours, you can make maybe a 24 amino acid peptide, because you have to do all the washings and everything. So something like that. Did I answer more or less?
Yes.
Say.
Regarding to the staples, 2 questions.
Yeah.
How many staples do you need more or less in average, and how do you work out how many do you need? So you probably do it in the 3D structure, right? Of the protein,
You
because
can do that.
otherwise, or the peptide, I mean, because you need to know where to put these.
Yeah. I mean, you can do different things. So let's go back to the staples so that everybody can see them. So we have them here. Right? One thing you can do is say, okay, I know this helix is going to interact with another protein. So if you put the staple in the phase that's interacting with the protein, that's not gonna work, because it's gonna interfere with the interaction and it's not gonna work. So you have some some software that allows you to see these proteins, and then you can just see where to put the staple. Not always you get it right based on this rational design. So sometimes you just have to make the compound and see if it works or not. About the number of staples you need, it really depends. So sometimes it's okay with a single one, sometimes you need multiple positions to staple, like 2 or even. So it really depends. And sometimes you can staple between 2 groups, sometimes you can do a massive staple between this group and the next one. So you can see, sometimes you can do rational design, so just look at the protein and see if it's okay. Sometimes you just have to make the compounds, test them, and see if they work or not.
And what are these staples made of?
Different things. So the original ones by Verdine, they were just hydrocarbon chains. So that's just chains of carbon and hydrogen. So chains of carbon with some hydrogens, and they have a double one in the middle. So you can make them of other things. These ones are very lipophilic, very fatty, let's say. So sometimes they're not good. So you can use other things. You can put oxygen atoms in there. You can put nitrogen atoms in there. You can do different things, but it's based on carbon, and also sometimes some oxygen, nitrogen, etc. Okay. Hey.
Well, I was a little bit late, so apologies.
Yes, okay.
I wanted to ask, can you explain a little bit more about any target that you are actually tackling that is not confidential, and give us an example. With this, I don't know, this enzyme, we are getting this peptide.
Okay, so can't really name names, but I can show something. So the typical case people deal with is this one, in which the interface between the proteins is a helix, right? And in this case, what we do is we make this helix and we try to staple it to make it stable. So that's the thing we try to do.
Yeah, but then you like
Yeah,
you have to kill us after telling us.
Yeah. I couldn't say the target. I mean, I can just say so. The typical ones in the literature, which we may or may not be working in, so P53 and DM2 is a typical one. So that's one of them. But many others.
So how efficient is this process in practice in terms of you have this protein-protein interaction that are mediated by this phase? What percentages of cases you will have a successful interference in protein interaction?
Okay, so that's a good question. So when staple peptides started a few years ago, say maybe 5 or 6 years ago, something like that. So they were going to be the solution to everything, right? So these guys just show these helix and say, yeah, put a staple here, it's going to be more stable because the protease is more degraded, it's going to be so helical that it's going to interact perfectly with the protein. It's not quite like that, so you have to try quite a few staple positions, you have to be really careful with your design with the computer, and then a lot of times it doesn't work and requires the synthesis of a lot of peptides and the testing to see if they really bind to the protein. So stapling is not always the solution. We are starting to see, but it is a solution for some cases.
Is there any holy grail in the literature, in the public literature of protein-protein interaction that needs to be addressed? Because they really can't at the moment using staple peptides.
That really can?
Is there protein interaction that if people have been trying hard to work?
And they don't manage. Yeah, there must be some, but I guess when they are unsuccessful, but well, there's been a lot of controversy. So there was this paper with this guy saying, yeah, staple peptides work really well with this protein-protein interaction. I don't remember which one it was. It was BCL2, I think. I don't remember exactly. So they were saying that staple peptides were working really well. They were basically doing what I've shown you here, disrupting this. Then these other guys publish a paper, basically not on their own research, but basically saying that the other paper was bullshit, saying that it didn't really work, blah, blah. But then the other guys came back saying that they didn't. So there's a lot of controversy still. Personally, I think staple peptides can work in some cases. We still don't have the holy grail of how we have to make them so that they work always. But they work in some cases. They're not awesome as they sell them to us, but they can work. But that's just personal opinion.
And are they specific as antibodies, for instance? How do you
depends.
check that they're
The specific.
specifically that protein?
You can do different things. I mean, yes. I mean, my company collaborates with this order called cell zone, which you can. That's not for peptides in general. You can just probably you're better than me, but that's more biology. So that's my field. But you can check with which proteins of the cell is interacting. Is your substance, your medicine, in this case a peptide, interacting? So you can see if your peptide is hitting only this protein, or if it's hitting a lot of other proteins that are similar. So that's a way to see. But there are other things. That's more the biology bit, which I'm getting familiar to it. Not so much.
And to test the permeability of the peptides?
To test permeability. That's a very interesting one as well. So yeah, to see if something gets into the cell, I mean, you're not going to see it with your eye because cells are small, and also the peptides, you're not going to see them. Yeah. So what people have been doing is putting there's this fluorescent tags.
Yeah.
They just take this peptide and say, okay, I'm going to put up something that's fluorescent.
Yeah, exactly.
And then with a microscope, if it gets in the cell, I'm going to see fluorescence inside my cells, right?
That is not affecting the structure?
Absolutely. So yeah, that's affecting because you're modifying your molecules. So what you are testing is not your original molecule, but it's something else. So now we try to make these fluorescent tags smaller. We try to use other methods to measure the permeability that don't involve modifying the peptide, because the moment you attach something, fluorescent or not fluorescent, the properties of the molecule change. So then so yeah, we really have to be careful with the literature when they tell us, oh, this peptide is really permeable. That's okay. We have to test it ourselves. And then
So could you say a bit more about why they're not immunogenic? Because is it because they're short sequences?
I think it's the size. So my idea is that it's the size. I mean, they're not as big as antibodies. But in general, they're not immunogenic. When you go to relatively large peptides, yeah, you have to be careful with this immunogenicity. But in general, they are smaller, so they tend to be fine in this respect.
Because then it would be nice if you could develop antibodies to the peptides if you wanted to visualize them.
Okay.
To see where they went.
Yeah.
You know?
Yeah. No, that's true. That's
That
true.
might be a disadvantage.
Yeah. That's true.
So concerning the non-immunogenic thing, so do you know if you have side effects, other side effects not concerning in the immune system? Maybe because of the dose that you have to do, because sometimes you find something that target what you want,
Absolutely.
but the dose is very it's very big and you kill the person. So that happens a lot in biology. I mean, when you are testing drugs.
That's true, but it's not only true for peptides.
Because you don't kill people, but I mean, you kill much. But that's the
You're absolutely right. So that's true for not just for peptides, but for any medicine, right? So you're testing it in vitro, you see that it disrupts this protein-protein interaction, okay, that's promising. Then you test it with cells, you see that it does all right, sometimes not. Then you test it with an animal, and something happens, it goes wrong, and it does something bad to the body, it goes to another organ, it stays in the body forever, and then causes something bad. So yeah, that can happen. So we have to treat them like any other medicine that you're trying to discover.
So it has, but you don't know if you compare it with other drugs, if it has more toxicity or anything.
So that's a good point. So peptides, if you use mostly natural amino acids, here they are not natural, sorry, proteinogenic amino acids, because so the amino acids we have in our proteins. So these are recognized by our body. So these ones are not toxic. So if you use mainly proteinogenic amino acids, then the peptide is unlikely to be toxic. It can be, but it's less likely than if you make one of these typical medicines that we have now in the pharmacy that we use something totally we make. So we don't know. They've been tested. These ones are fine, but before they get to the pharmacy, then
So there are many studies concerning this using animals, for example, that they arrive to that point that you know that it's working in cells and everything, and they arrive to, or maybe not.
I mean, I haven't one yet, but I'm sure there are. It's like any other medicine. So a big problem in pharmaceutical industry is something called attrition. So that means that everything is going really well. The peptide is doing what it has to do, even in cells, even in rats, even in other animals. And then when it gets to humans, it's alright at the beginning, and then there's a side effect when they're testing it. But they do it in very small doses, so they can really monitor it. That can happen like with any other medicine. It's the same.
So how many of the medicines that we are using now are based on peptides?
So I don't know the absolute number. I know that this about 25 billion pounds, is about 10% the amount of the money generated by pharma industry. So it's about 10%. That might not seem a lot, but you just have to think that it's not 10% of effort. What pharma industry is putting in peptides is much less. So it's just increasing now. So one could think that if they put more effort, if they crack the cell permeability, then these numbers can increase.
How expensive or cheap is this to develop medicine based on this?
Okay, so peptides are always going to be a little bit more expensive. They're going to be usually in between antibodies and peptides are going to be in between small molecules and antibodies in terms of price, but that's a generalization, so it won't be true always. So they're larger, so they're going to be more expensive, but they're getting cheaper and cheaper. So the building blocks are getting cheaper because, I mean, the more demand there is, the cheaper are the building blocks. They're more expensive, but I think they're getting cheaper. And also, the thing is that if it's your only option to treat a disease that involves a protein-protein interaction, you just have to go for it. And it seems that with the small molecules, the typical medicines is going to be really hard to target on this involved.
Is there a competition now in the market between like companies who want to create drugs against, like with using peptides, and other companies who are using antibodies, and other companies that are using small molecules? Are they all targeting the same kind of molecules and protein interactions?
I think that is kind of, I don't know, in global, I don't know. But I would say, this is a tricky one. I think, for instance, antibodies, they're really good at modulating protein-protein interactions, but in general, they don't get into cells. So it's really hard to get an antibody into a cell. Whereas with peptides, people in pharma industry are confident that they're going to manage to crack it and to get peptides into cells. So for intercellular targets, it seems that this space is going to be more for peptides. For extracellular targets, my point of view again, I think antibodies are winning. So it's a generalization. There's an important biopharma antibody unit as well. More important than us. So it's larger than us. We're getting there.
Have companies, pharmaceutical companies, got really closer to targeting peptides to specific organs or cell types?
We're trying, but yeah, we're trying.
No, yes.
I don't think it's that yet, but I think it'll be that in the next years.
Because that's sort of, I use peptides in my PhD,
Okay.
but I'm a biologist, so chemistry is good.
Yeah.
But that's one of our concerns. Is
Yeah,
you're targeting
absolutely.
an interaction that's in every organ.
Yeah,
You want to
yes,
hit the
absolutely.
brain. Yeah, it's just.
It's something that's, you're right, I think it's something that's very important, and I think it's something that's going to happen in the next years.
And how do you solve that problem with the peptides that are in use now? Just hydrodoses to get the
So I will have to look at each particular case probably. So what do you mean?
because if this peptide is able to cross every membrane, every barrier in every cell, in every organ, every tissue, obviously the amount reaching the target in the real
It's gonna be
tissue is
smaller.
be
Absolutely.
much
Yeah,
smaller.
yeah.
So what do you do? Just super high dose?
No, I wouldn't go for super high doses is the typical. Yeah, let's go for it, but it's not the solution, I think. So high doses, what they generate is side effects. So the medicine does many other things. So formulation can be a solution. So formulation is like, okay, would I have this as a capsule? Would I have this as a tablet? So as an injection of this type, injection of this other type, that could be a solution.
But anyway,
Other
injection
things.
or oral is
Oral is really tricky for peptides. So oral peptides is difficult to get them oral.
it's a nice environment. So
Oral is possible, and the cyclosporine is oral, for instance. And it's oral thanks to the formulation. It's a specific formulation. Cyclosporine, this cyclopeptide I was showing you here, is very lipophilic, so it doesn't have nothing similar to water on it. So it doesn't dissolve very well in water. But they've made kind of a formulation with an oil or something like that you can just take a pill and it works. So sometimes with the formulation you can do it, sometimes you can't. So oral peptides is challenging for now.
In the pharma industry, when you're synthesizing peptides, and trying to find, let's say, a peptide that targets a specific, do you use higher structure-based approach, or do you just do a huge phenotypic screening of a large library?
We do both actually. So so we do both. So something I haven't said here because I was talking more about the chemistry of peptides is that you can make peptides biologically as well. So with some organisms that just make them, and you can generate these massive libraries of peptides in smaller amounts. but you have a massive library of a massive collection of peptides, and then you can just test it against your targets, get some hits, and then you ask the chemist to make them or to improve them or anything. So that's, yeah, we do it with big libraries, but with some targets you can try with this rational design. Is looking at the protein, looking at the peptide, and see what you can do.
Would you say that these peptide medicines in principle are not good for brain diseases and retinal diseases? Because they need to cross the blood.
Yeah, I was going to.
I'm not sure if someone asked me before because
Yeah, sorry. Yeah, no. I was just going to say that they're not good because they don't cross the blood-brain barrier. Yeah, so that's this barrier that the brain has to protect itself. But yeah, it's going to protect itself from bad things, but also from medicines. So yeah, that's a tricky one for us. Sorry?
the touch ones do.
The touch. Yeah. So it can do. I mean, a friend of mine, yeah, you know, yeah, so that's good. But are there many peptides in the as medicines? I know there are a few in the clinic.
Yeah, I think there's only two that are in the clinic and it's very private, but I work in animals and I've seen it in the brain, so it does come in.
Okay. So I've seen the review in which there are some of the peptides in the clinic, and we have some of the TAT peptides, but I don't know if they will make it to the end. You know, a friend of mine was doing a PhD, was just testing if these TAT peptides and other so-called cell-penetrating peptides, they were crossing this blood-brain barrier. And I didn't remember exactly the results, but if you say it.
That was actually my question.
Okay. That's good.
Well, I have one more. So I'm a
Okay.
computational chemist, and I'm
Okay.
doing structure-based design on peptides actually.
Okay, that's
So
good.
that fits quite handy. Did someone ever play around with post-doctoral modifications of these peptides? Are there methylated peptides out there like this place phosphorylated or whatever you want?
I mean, people have done it, yeah. Yeah, I mean, let's start with methylated. Something that's been used is to methylate. I don't know if you mean this kind of methylation. Maybe you mean more in the side chain, glycine. Okay. People have done it. I haven't done it personally. I've seen people doing it. I haven't done it myself. Yeah, people use sugar sometimes to just. Sometimes they help getting going to the right type of cells, but that again, that's not my field, so I don't do it. But some people do that. What we do as well is we methylate. We put a methyl. A methyl is a carbon on 3 hydrogens. We just put it in this nitrogen here that usually it has just a hydrogen. And sometimes putting this methyl here, it improves the properties of the peptide. It makes it maybe more greasy and then it's more cell permeable. Sometimes it causes other things. It causes it to fold in a different way. So that's what we do sometimes.
Any more questions? No? Okay, so thank you so much.
And I'm just going to give you some information about the next seminar that we are going to have in the Spanish researcher in the UK. And it's going to be, it's called Tribute to Juan Lins, who was a Spanish sociologist, political scientist that was working at Yale University and died last October. And well, this seminar will be given by Dr. Jeffrey Miley in the Department of Sociology, the 13th of April at 6:30. And he was a disciple of Juan Lins, and he will talk about the work of Juan Lins, that is basically about totalitarianisms and authoritarian governments and also transitions to democracies. And I hope you're going to enjoy it because Jeffrey will give us all the what Juan Lins did during his studies and also how we can extrapolate this into the social, political, and economic crisis that we are living now. So you are more than welcome to attend to this seminar, the 13th of April. Okay, thank you.